So noswaith, good evening, everybody. Thank you for joining us for another webinar, one of our winter webinars. This one tonight, we have the pleasure of having Peers Davies, Dr. Peers Davies, I should put it properly, who's also a vet, first and foremost, talking us through some of the challenges we all have in our industry. I suppose it's, as always, we try and make these sessions as interactive as possible. So you'll see that there's a chat at the bottom of your screens there. If you have any questions as we go through, please just type them in there and we will endeavour to either answer them there and then, if it's appropriate for where Peers is at, or we'll save them towards the end. But if there's anything, hot on your mind, please put them into that chat. There's never a bad question, to be honest with you. It's always better to do it while it's fresh in your mind, rather than forgetting about them. So tonight is very much about health and avoiding disease and what to do with it when we have it. And Peers is very apt in giving us those guidances, because he does that for Innovis. And we depend greatly on Peers' wide knowledge base and also quite a pragmatic approach. And I think what you'll find probably at the end of this evening is that there needs to be quite a lot of pragmatism and a lot of common sense and weighing up of things when we go through these. Obviously, we do what we do as Innovis to try and protect customer flocks. The last thing we would want is to actually give anybody disease, because all the genetics and all the benefits that you'll see in your systems changing all really get negated away and they disappear if you bring in disease into your flock. And we urge and plead for people to do that with anything that they buy, to be very vigilant. But again, Peers will give us a much better view on that. So enough of me spouting on, Peers, over to you.
Thanks very much, Dewi. That's great. I'm just going to share the screen now and then should be able to everyone see it. Right, so hopefully everyone can see the screen. So thanks, Dewi, for a very kind introduction. So I'm a farm animal vet, come from a sheep farming background, and I've been working in farm animal practice for 15 years now. The last 10 years or so, really sort of balanced between consultancy and academia. So a lot of my time is spent on research and teaching vet students at the University of Liverpool. And one of my main areas of research is around production limiting diseases, principally the infectious production limiting diseases of sheep, so viruses and bacteria. So I'm going to give you a brief summary, really, of some of the key, we call iceberg diseases. Each of these diseases could really justify several hours on their own, but obviously that's not possible. So we'll try and keep to the sort of most salient points. And obviously, any questions, really good to have questions as we go along, just to make sure that anything which is of interest that maybe isn't covered in as much depth, we can go into in a bit more detail. Okay, so hopefully it's going to transition on. Doesn't seem to be moving.
It should, if you click on it, you should. There we go.
Okay. So hopefully now I can see the next slide. So the term iceberg sometimes confuses people. So what we mean by an iceberg in terms of diseases in sheep is really the difference between the clinical case, this extreme case where this animal is obviously visually ill as the tip of the iceberg. But for some diseases, there's actually a much larger number of individuals within the flock who are infected and either incubating the disease or suffering with what we call subclinical disease. So this is where it's not visually obvious to the farmer, to the owner, to the vet, but there's still a difference in the level of production because of the presence of that disease, of that pathogen. So that's the definition. And you could have an awfully long list of potential diseases that we could term as iceberg diseases. We focus in most of the time on really five diseases. And what we want to cover in this webinar really is those kind of key, most practical bits. So what does the disease do? How can we go about detecting it? How can we keep it out of a flock if we just identify that a flock is currently free? So how can we use appropriate practical biosecurity to keep it out. And that comes really down to pre-screening of stock and quarantine procedures. And then if it is in the flock, how can we deal with it? What control measures have we got to be able to minimize the impact or eradicate it in some such situations? Okay, so they're the kind of key areas we're going to try and deal with for each of the main diseases. Okay, as I said, there's a vast amount of detail about each of these diseases, far more than we could possibly cover in this webinar. So back in 2018, we wrote a manual on the options for management and diagnosis of these key iceberg diseases, really aimed at proactive farmers, vets and advisors to be a reference guide. So that was funded by HDB, and that manual is freely available on the HDB website. So you can find it through the website or through the link on the webinar. And that gives you a much more detailed, comprehensive breakdown for each of these diseases. Okay, so the 5 diseases we're going to be discussing tonight are board disease, CLA, so caseous lymphadenitis, mydevisner, ovine paratuberculosis, or Johne's disease, ovine Johne's disease, and ovine floral adenomatosis. Okay, now we typically break these diseases up really into 2 groups. So border disease sticks out as a slightly different disease to the others, in that the main presentation of the disease that we're worrying about is a reproductive presentation. So reproductive disease, abortion, small weakly lambs, very high empty rates when it first enters a flock. And that's quite different to the broad syndrome that we would associate with the other 4 diseases. So broadly, although there are differences between them, we're looking at the other diseases as chronic wasting diseases of ewes and rams, which will present as then that slow grinding reduction in productivity, eventually higher culling rates and reduced longevity, and also reduced fertility and fecundity as well, so reduced scanning percentages as they are in negative energy balance. Now, although we've listed 5 diseases, they're not all equally important, for want of a better word. So their level of importance you could argue about subjectively, but roughly you'd say, well, they're important if they're common, and they're important if they're severe. So of these 5 diseases, the 3 which I would judge as being the most important, the border disease, mydevisner, and Johne's, for that combination of the severity of effect within the flock and the number of flocks affected, and how far within a flock it will spread when it gets in. Okay, so before we go into the rest of the webinar, just a bit of a quiz question for you all. So which of those diseases do you think you have or suspect you have in your flock at the moment? So you can tick as many options as you like within the question. So we'll just give everyone a minute or so to answer the questions, and then we'll see what the results look like.
And I suppose, Piers, equally important, if you have no idea what's going on in the flock, which lots of people are in that boat, then obviously tick don't know. Yeah, and you won't be alone in that. I guarantee you there's other people that unfortunately don't know if they've got any of these iceberg diseases operating. And if you're very, very fortunate and confident, you can always tick the none of the above, which is obviously very, very good. And Laurie, who's operating this, won't let me put anything in there. Otherwise, I'd be ticking away for you. Takes a bit of thinking, doesn't it? And I'm sure Piers will go into these diseases in terms of their symptoms and elements as we go through the presentation next. So although you're ticking now, you don't know, you may well in a few minutes time or half an hour's time actually put some dots together and say, actually, maybe I do have something like this here. So there we go.
There we go. So yeah, the don't know category the biggest single category. So that's quite important and relevant. So we've done a number of studies over the years, and that is always the biggest category. It's got smaller over the years, so people now are getting a better and better appreciation of what is in their flocks. When we first did one of these studies in 2013, basically the don't know category would be almost everybody. So now at least we're getting a little bit more awareness of the presence of the different diseases. So I'm just going to close that now, and then we'll go on to the next section. So there we go. So do we need to worry about them? You know, are these diseases actually, are they around very much? Are we seeing a lot of them or not? So we've done a number of studies over the years, and there's quite a lot of published literature for some of these diseases and not so much, which allows us to get an idea of the proportion of flocks in the UK which are affected. So as you can see in red at the bottom, we've got border disease and Johne's. So by far the most common of those 5 diseases are border disease and Johne's, with the majority of flocks in every study that we've run turning up positive for Johne's. And interestingly, it's a very extreme example of that poll that we just ran. In every study, we find that the vast majority of people who test positive, flocks who test positive, didn't know they had it beforehand. So it's a true iceberg disease in the sense that it's very common. It's a very big below the surface level of disease, and a level which is above the surface, which is invisible to most people. And we'll talk about that in more detail in a bit. But if we go back to the top of the list, so mydivisna is an interesting one. So obviously everyone knows we've got a mydivisna accreditation scheme. And you could argue that actually that over the decades that that's been functioning, it's been pretty successful in slowing down the rate of mydivisna transmission. But we are now in a phase where we're seeing the level of infection within the national flock going up pretty rapidly. So projections of around 10% have been discussed, and that is looking to be more and more likely. And there's quite a regional variation within that, again, which we'll get onto later. Then other diseases
Sorry, Piers, can I just interrupt for a minute? Sorry, I told you to use the chat button before. I have been corrected by Laurie. It's actually a Q&A button on your screens at home. So it's the Q&A button. If you've got any questions, please use that. You don't have a chat button. Sorry, Piers, just a formality.
No worries, no worries. Yeah, so we've got this sort of very different pattern in terms of the relative commonness, the relative prevalence of these diseases. Okay, so how can we go about detecting them? Well, the single most useful group of sheep on the farm, in any flock, for diagnosing iceberg diseases are the cull ewe. So cull ewes, either for sampling live with blood samples or are very likely to prove these diseases, but also cull ewes by post-mortem is incredibly useful. So over a number of years, we've been working very closely with farm post-mortems with Ben Strugnell, and we've done a lot of cull ewe screening within the Innovis breeding partners, and also wider as well, in association with HDB, the different programs across England and Wales, looking at cull ewe screening. And this is incredibly useful as a way of pulling out not just the iceberg diseases we're talking about here, but actually a whole range of other diseases as well, other abnormalities, and really being able to see if the proportions that you're seeing in a flock is what you'd expect or higher or lower than you'd anticipate. And there have been some very interesting findings out of this. And this was the first study where we were able to estimate Johne's prevalence nationally by comparing flocks across different areas and from different sources. So either from the HDB demonstrator farms all the way across to individual commercial flocks who were participating as well. So it's a really, really useful process, but it does mean that you're sacrificing the cull ewe value. You're sacrificing that value in order to get what is probably the most valuable diagnostic information you can. The alternative to that is using blood samples. And fecal samples, blood samples, very useful, very straightforward to get a picture of mydevisner, border disease, and CLA. The tests are not perfect. Some are better than others. So, for example, the mydevisner and border disease assays are a bit better than the CLA assay, CLA blood test, but used in the appropriate way with an appropriate number of sheep samples, you can get a very useful understanding of the level of the disease in the flock, or you can get a good level of confidence that the disease isn't there. And that really comes down to those characteristics of the test, how good the test is, and the number of sheep that you're sampling. And the groups that you sample are subtly different between the different diseases. For example, with border disease, we're generally going to sample the lamb cohorts, whereas for pretty much everything else, we're sampling stock rams and culls. Okay, fecal samples are also very useful. So they're by far the best way of diagnosing yonis. The blood samples for yonis are not very good in the sense that they're not very sensitive. So an animal will only develop antibodies to yonis right at the very end of the disease, which is not really what we're interested in. We're interested in picking it up, even if it's there, that subclinical phase. So we do a mixture of testing, culls, blood samples, and fecal samples to establish flock status and also individual status in some situations, which we'll talk about in a second. Okay, so go on to border disease to start with. So it's a nice example of an iceberg disease. And your iceberg, your tip of the iceberg, are those classic hairy shaker lambs that everyone thinks of. And the mistake is to think, oh, well, I don't really get any hairy shakers, so can't be such a bad disease. But the real issue is actually when you have the virus working its way through a flock, particularly during tupping. So if it enters with a purchased ram or with purchased females, typically they're going to be mixed pretty soon after arrival during tupping. You're going to get then a lot of transmission of the virus through that tupping period. Now, that just the fact of getting infected with a virus of that type means you're going to have an increase in temperature, you can have a fever, and that fever can be enough to stop them from conceiving. And that can be associated then with very, very high empty rates. So the sort of bulk of the iceberg then is that reproductive impact, those lost fetuses, those high numbers of empty ewes, rather than the peak of the iceberg, those few affected hairy shaker lambs. So the way it works is actually very, very similar to BVD in cattle. And the two diseases, the two viruses are so similar that they can actually jump between different species. So every year we'll get abortion storms in sheep associated with BVD from cattle. And it can go the other way as well. Border disease can go into cattle. And basically you've got this pattern where infected sheep, infectious sheep, can then spread the disease, come into a new group. If they infect ewes before tupping, and the ewes have a couple of weeks in order to be exposed and generate an antibody response, then those animals can be quite happily managed with no issues. They develop an antibody response and they're not troubled by it. However, if they're exposed during the relatively early phase of pregnancy, they can just fail to conceive, so you get early embryonic loss and they turn up as empties. And then you can later get these PI, persistently infected lambs, produced. Later in pregnancy, when the lamb has already developed a functioning immune system of its own, the lambs can actually fight off the infection if they're only exposed quite late on in pregnancy. The problem we have with border disease is actually it's an issue because it's not that contagious. If it was very, very contagious, then actually it would burn its way through a susceptible population very quickly and die out. The problem we have is it's not that contagious, so it takes a long time to work its way through a population. And that actually makes transmission between flocks more likely, and also transmission between generations within a flock likely. So those very, very contagious viruses actually, in some ways, are less of a problem. But this sits in an awkward middle position. We don't have any licensed vaccines for the disease in the way that we have for BVD in cattle, although occasionally the BVD vaccines are used off license, are prescribed off license for use in sheep. So that has been reported. That is occasionally done in certain circumstances. We're just not really sure how effective that is. The traditional approach is to identify if there's a problem in every group of purchased replacements or rounds. If they're diagnosed that it is there and it's circulating with a combination of antibody tests and tests directly for the virus, then we can make some decisions. Either we don't keep those animals on farm, or we do keep them, but we keep them isolated in their own group till after lambing, so they don't spread it to the rest of the flock during that critical pregnancy period. And after that period, what's commonly done is to mix them very, very thoroughly to try and get transmission of the virus through the main flock before the next tupping period, although that is quite hit and miss because the virus often isn't that transmissible. So it's not a perfect solution. What's sometimes termed natural vaccination approach, but it's not perfect. So we've got this very common disease. A third of farms already infected, so that's quite a high chance of actually passing it on. We've got good
Piers,
tests.
sorry, Piers, I've got a question from Sam, just asking, just before we go off it there. Would we get low growth rates in infected lambs?
Yeah, so I think my camera's doing something strange. I'm just going to stop my video because it seems to be having problems. So how does it affect growth rate in lambs? So that's a really interesting question. We've got some slightly conflicting data there on border disease in lambs. We've got a couple of studies which have shown border disease decreasing growth rate, but we've also got a group of lambs where we see an improvement in growth rate associated with border disease. Now, reconciling those things is not necessarily that easy. If we extrapolate from BVD in cattle, we know that it has an immunosuppressive effect. So in calves, that's a massive problem when we're rearing, particularly dairy calves, all in a close proximity to each other. So that immunosuppressive effect makes them more susceptible to pneumonia and has all sorts of negative effects there. The only way that we can think how you could get a positive effect on growth rate, which has, as I say, been demonstrated once, is actually because in certain circumstances, suppressing your immune response can be advantageous for lambs, particularly where you have exposure to gastrointestinal roundworms, where the part of the problem the lamb experiences is its own immune response to the challenge. So if you damp down their immune response, they can be subject to less of a growth check because of the worms. Now that's very much a hypothesis without any proof one way or the other, but it's quite difficult otherwise to reconcile the conflicting results that we have seen on growth rate in lambs. That's possibly a longer answer than you were expecting to that question, but it's an interesting area.
Thank you, Piers. There's one other question come through from Maddie saying, just to clarify, how is the virus actually transmitted, Piers?
So that's a good one. So it's transmitted like an awful lot of viruses. So you've got orofecal, so in the feces, but also in droplets from respiratory secretion, so droplets from the nose. So it can be spread in both ways. We think probably that the spread through the air is more important, and that's similar to what we see actually with BVD when it spreads from cattle to sheep as well. So you don't need direct contact with the same ground. The same airspace is sufficient to give transmission.
Thanks.
Okay. So we've got two different types of tests that we can use. We can use antibody tests to check if a group of animals have been exposed to infection. And we also got the antigen test, which actually picks up the virus itself, much like we're all familiar with COVID tests. Now, in the context of sheep biosecurity, one of the most important things is to identify any persistently infected animals which are continually shedding virus. So for that reason, all rams that Innovis sells actually go through a PI test. So they test to make sure that they're not, there's no virus present. And that's a pretty important test for anyone, really. If you're running a flock, then whilst those rams come into quarantine, then checking that they're not PIs is a very important step. You would be very unlucky to find one. It's a very low proportion of rams who are PIs, very, very low. But the consequences of missing it are also very, very high. So whilst the vast majority of the time they'll test negative, the importance of actually making sure you don't bring it in is very high. The bigger risk is actually trading large numbers of females. So large numbers of ewe lambs represent a bigger risk, really, than others. And there, what we frequently do is we'll test a portion of the group for antibodies, and that shows whether the virus has actually been transmitted within that group. If there was a PI there would definitely be some animals there with antibodies. And that's a more cost-effective way of dealing with it when you're trading large numbers of female organisms. The number of animals that you test really comes down to the size of the group and also how confident you want to be find it. So that's not a simple answer of, oh, just do half a dozen. That's almost always going to be the wrong answer. But it's a question to have with your vet to decide actually what is the level of confidence that you need to have, and therefore how many animals need to be tested. Okay, so moving on. So moving on to CLA. So CLA is probably of these diseases the one which has got the least evidence of a major production limiting effect. So we have these very obvious visible lesions, which are the lymph nodes, which sit behind the jaw. We don't have a lot of evidence for other production limits from some studies from Australia, which show an impact on wool quality. Now, obviously, wool is a really good indicator of the general physiological health, the efficiency of those animals. So if you get wool quality, it's a reasonable assumption that actually there's going to be other impacts on the animal's efficiency. But we haven't actually got any explicit data on impacts on growth rates or milk yield or other performance indexes. It's obviously been targeted in certain breeds and in certain populations because these very visible lesions are obviously a problem for sales. But it is it's not one that we're overly worried about in terms of production limiting effect. It's also not particularly common in the national flock, more common in certain areas, but there are issues that you can have with internal abscesses as well as these external abscesses. So those little lumps behind the jaw, the lymph nodes, can be mirrored by much bigger abscesses internally. So on the picture on the left, you've got what are called mediastinal abscesses. So these are between the lungs. So you can see this huge, great big cheesy lesion there. Obviously, these are extreme cases. These aren't necessarily typical, but these are sort of worst cases. And you'll get them in the guts, and you can also get them in the udder as well. Okay, so we've got a relatively small portion of flocks who are infected, more in particular breeds, particular groups of breeds. But we do have a test which is available and which is effective enough to allow eradication with a test and cull approach. So that has been shown to be effective. It does take multiple rounds of testing them. So approximately 5 rounds of testing in order to get eradication. You've also got the option of a vaccine. So the Glanvac vaccine, which we typically import from Australia and New Zealand, is effective and has been widely used in many flocks in the UK, either permanently or just intermittently until the disease is under control, potentially eradicated. So Innovis obviously screens flocks for antibodies, and any animals with lumps get tested by swabbing those lumps, the internal contents of those lumps, and try to culture the bacteria. So that we have a good screening approach for that, and as I say, it's a very, very rare issue. So
yes, sorry, just a quick one just before you go off CLA. Richard's asking, are there 2 types of CLA? Is the CLA different here to the one that happens in Australia?
Yeah, so it's a really good question. So this has been a subject of significant debate. And to be honest, it comes down to how different is an important difference. So what we term in epidemiology, the discriminatory power. So simply because the sheep populations in Australia and New Zealand have been separated for some time, means that the diseases and the pathogens associated with them are also diverging over time. So yes, they are inevitably different. Are they so different that the vaccine doesn't work? That doesn't seem to be the case. So where you have flocks with a lot of lumps, a lot of disease, they start vaccination, we see very good clinical efficacy. So we see the proportionality of lumps really drop very, very rapidly. And that's only going to happen if the strains are susceptible to the vaccine, and the vaccine's developed on the Australian and New Zealand strains. So we have a similar debate around the Campylobacter vaccine and the strain associated with that. So we have to really look at what happens in the UK when they're used to see whether we think they're efficacious or not. And the clinical reports of their use really strongly show that it does work as a way of reducing or preventing disease. So yeah, again, a slightly grey area answer, but hopefully useful.
Thank you, Piers.
Okay, so moving on to Mydevisna. So Mydevisna is a bit of a different one. So you get this massive swelling of the lungs. So on the left, you've got some normal lungs there, relatively small. You see the heart there. On the right, you've got particularly the left lobe, the left side of the lungs of the set on the right. You can see they're massively, massively enlarged. And what that enlargement is caused by inflammatory fluid, so white blood cells, being filling up the spaces between the alveoli, between the air sacs. And this makes the exchange of gases across the lungs much, much harder, less efficient. And this reduction in efficiency is what then drives the animal down. So lung efficiency is incredibly important for the animal's normal metabolic functioning. Sheep are actually very good compared to other species at coping with quite a lot of lung disease before it really starts to affect them. But Mydevisna can be really very severe and will lead to emaciated, but visually obvious. What happens before they get to that point, though, are reductions in milk yield, particularly in ewes. So we've got some good data, both from Europe and also from the UK, showing 8–9% reductions in milk yield in dairy ewes, where we can measure their milk yield very easily. That doesn't necessarily mean we're getting the same percentage reduction in milk yield in suckler flocks, but it certainly would be reasonable to assume that there is going to be some reduction in milk yield and therefore lamb growth rate. And that would be supported by what we see when we find big commercial flocks, suckler flocks, where Mydevisna is diagnosed, and we can track back over time to look at their performance. So in quite intensive flocks who do a lot of creep feeding, what we see is the amount of creep per lamb to get lambs to finish starts to creep up and up and up as the Mydevisna prevalence within the flock increases. So as the larger and larger number of ewes become infected, more and more of the lambs are basically being suckled by ewes who are producing less milk, is the hypothesis. And that means that the proportion of creep that needs to be fed goes up to get those lambs to finish. And obviously with the price of creep at the moment, that's a very immediate impact on your productivity. And the same is going to apply to forage-based systems as well. You're going to end up with a reduction in growth rate of that critical, most important growth rate early on in life, which is by far the cheapest. So if that's being reduced, then you're going to have problems later on. So there are some of the issues we see with Mydevisna. We do have very effective screening tests for Mydevisna, multiple different blood tests with different attributes, some better for screening, some better for diagnostics. They're different things. So we use different tests when we have a population where we think the vast majority are going to be negative, and we're trying to find a few positives, compared to when we're trying to discriminate between individual animals, positive or negative. So we've got good tests available, and it's a case of how we use those to identify problems. How common the problem is, I mentioned before that it's getting more common. The map on the right-hand side is a few years old now. So it's based on quite a unique data set, which gives us a really interesting insight in myelofibrosis, which we don't have for the other diseases. And this comes about because across the UK, we collect blood samples from random flocks in order to prove our national disease freedom for a different disease. So to prove our national disease freedom for brucellosis, we take these random samples. And that means there's blood left over. So what's been done several times now is when these bloods are collected, the spare blood is then run on the mydevisna test to identify the proportion of flocks across the country which are infected with mydevisna. And the money is spent on mydevisna because the tests available are the most accurate compared to the other diseases. So it's the best use of limited funds. And what that's shown is that you have this very patchy pattern with certain areas, up to 15% of flocks infected. And as I say, this is a few years old, so the situation's worse now. So more areas are showing positive for mydevisna, and a larger number of flocks positive within those areas. So it is an area of increasing importance. Once it gets into a flock, mydevisna is serious because it can carry on going, spreading within the flock until you get to a very high proportion of ewes infected. And that means that those production limiting effects are spread across a far greater portion of the flock. Okay, so similar to mydevisna, so another quite closely related virus is Yargziti, which causes ovine pulmonary adenomatosis. Unfortunately, we don't have some of the good aspects of mydevisna, so we don't have reliable lab-based diagnostic tests in live animals. And because we don't have those good lab-based tests, we don't have good estimates of how prevalent it is. We've got some estimates which have come out of fallen stock post-mortems and out of abattoir surveys, which indicate that it's not very prevalent, very low percentages. And we don't seem to have what we get in mydevisna, which is this spread to very high proportions of the flock when flocks do become infected. It seems to be a lower, more chronic level. We don't have any prospect of a vaccine for OPA, similar to mydevisna. We don't have any prospects of a vaccine there either. In terms of tests, we've got the possibility of using ultrasound, which is probably an acceptable test as a diagnostic tool. So when you've got a bunch of ewes who've got symptoms, so they're thin, they've got respiratory signs, so they've got increased breathing effort or increased breathing rate, you've got reasons to think these animals may be suffering from disease. It's a fairly good test in that context. But if you try and use it as a screening test, then what you run into is the fact that it's not sensitive enough to pick out all of the animals who are infected. And it's not specific enough to rule out other things which can look very similar. So you don't really have the characteristics of a test that we would deem typically as being acceptable as a screening test. So if you want to use them that way, you have to really practice some extreme caution in what you expect to get. Okay, so now moving on again to a disease which is much more common, and again where we've got some better data and some better ways of managing it. So ovine Johne's disease, so we had a couple of respondents, few respondents who identified they had Johne's in their flocks when we did the poll earlier on. This is a one which really interests me because you've got this massive proportion of flocks in the UK have it, and yet almost no one knows they've got it. So in that sense, I think it's a very interesting disease. when you compare it to what we see in cattle. So in cattle, Johne's has been well discussed for many, many years. And you can talk to farmers who've known they've had Johne's disease in their cattle, managed it in their cattle, and never even realized that sheep could get the disease. And part of that is, what does this tip of the iceberg look like for Johne's disease? The tip of the iceberg for Johne's disease doesn't look like the tip of the iceberg for cattle. So typically they don't scour. So people have assumed that, well, if it's not scouring, it can't be Johne's disease. But sheep don't behave in that way. So they will lose weight and they will develop clinical signs which are often attributed to other causes of weight loss and never investigated. But the impact on productivity, as we'll see in a second, can be quite dramatic. Okay, so the way it works is very similar to calves. So lambs are infected with the pathogen, which is MAP. So this is Mycobacterium avium paratuberculosis, MAP for short. So that they're infected early on in life. And this is really important. You can't infect a sheep with the bacteria later in life. It depends on getting in when the gut is still developing. Needs that developmental stage to get in. Okay, so once it's in there, it's going to start multiplying, and that'll take some time. It's quite slow. So over 2 to 4 years, it'll multiply. And then several different things can happen. So either the sheep will be quite resilient to infection and not show any disease at all, just shrug it off. That's the ideal best case scenario. We don't know how common that is. That's going to be partly genetics. And we know that genetics plays an important role in susceptibility to Johne's in all species which get it, including humans. There's also a group of sheep, and this is the tip of the iceberg, who are highly susceptible, and they develop very dramatic, aggressive clinical signs, become very thin. Some of them will scour, but the majority of them won't, and they'll die on farm. They're the tip of the iceberg. But there's then a big group who are moderately susceptible. And these are the ones that I'm most concerned about. These are the ones which develop production-limiting disease. So what happens is they start diverting energy towards their immune system to try and fight off the infection. So they're diverting energy away from storing it as fat. They're diverting it away from other things like their reproductive system. So when they're fighting off the infection, they're actually causing inflammation in their gut as they're trying to kill the bacteria. And that inflammation then makes their gut less able to absorb nutrition. So they're actually getting less energy in now than they were before. And they're using more energy to fight the infection. So the ewes start to lose body condition. And this can be quite subtle to start with, but as they start to lose weight, as they enter negative energy balance, they're diverting energy away from other body systems, particularly their reproductive system, in order to conserve energy. Now, those ewes are then also shedding the MAP bacteria back into the environment and infecting the next generation of lambs. And this reduction in fertility, this reduction in the reproductive system, means they're more likely to turn up as empties at scanning. And then those empty ewes are being culled and replaced, but they're not being investigated, which is why people aren't realizing in the main that Johne's is there as an issue. Okay, so this is driving these higher empty rates, which we've now shown in flocks which have Johne's compared to which don't. So if we think about what that results in, if you're culling more ewes because you're getting a higher empty rate, then you're also replacing more ewes. So what we see is this younger age profile. Excuse me. So infected flocks in red, you see that they've got a higher proportion of the flock, so 0.3 or 30% of the flock in that youngest age category, so year 1, and then they start to cull more in year 3, 4, and 5. So the proportion of the flock in those older age groups drops compared to the blue line, which are the uninfected flocks. So this is a big effect on our sustainability. It's a big effect on things like carbon footprint as well, because these ewes aren't having the length of productive life they should be able to achieve. So this longevity effect costs us in many different ways. So when we compare these groups of farms, these Johne's infected flocks versus uninfected flocks, you're looking at average median replacement rates of about 30% in infected flocks versus an average of 25% in uninfected flocks. And that's a big difference. Obviously, it's not true for every flock. Some flocks, for other reasons, might have higher culling rates or lower culling rates. But overall, that's the pattern that we're seeing. So it is a disease we can tackle, though. So there is a vaccine which is available, been around for quite some time. Very heavily used in Australia, used to some extent in New Zealand, and used quite heavily in Spain as well, where it's first developed. So it's not a perfect vaccine. It's quite an old vaccine, so it has some drawbacks in terms of developing quite a strong reaction at the site of injection. So similar to some of the problems we get with footbacks, but it does have the ability to reduce the shedding of bacteria and reduce the proportion of animals who suffer production limiting disease. So we can't eradicate the pathogen because it lives so successfully in the environment, but we can reduce it to the point where we're infecting fewer lambs in the next generation, taking the pressure off in that way, and also directly taking the pressure off the animal by giving it a more effective immune response to the bacteria. So it's probably a candidate where we'd expect that we'd get better results with a next generation of vaccine, but we've got decent tools available now to control the disease. And we've got good data from Australia, particularly on the cost effectiveness of it in their systems, where you've got less productive ewes anyway. So it might be reasonable to expect with higher levels of output that the efficacy and cost effectiveness would be higher over here. So that is one area that we're currently working on as part of our research work to try and understand the cost benefit of both vaccination for Johne's, but also for some of the other diseases that we've been discussing tonight. Okay, so almost over. So this can feel a bit overwhelming. It's an awful lot of information, an awful lot of diseases. All the diseases are a bit different. So what do we need to do as a sort of step-by-step approach? Well, the first thing is to work out actually what is your current status in your flock. Some of these things you're not going to have to worry about, others you will. But you need to establish that first rather than putting them all into a box and not wanting to touch the subject. So establish that with your vets. There's some fairly straightforward ways we can screen for these diseases, which are appropriate. Numbers of animals tested will depend on the size of your flock and how it's structured. Have that conversation, and that'll give you a starting point in order to deal with it. So keeping out things that you currently don't have and managing things that you might already have. Okay. Similarly, when you're then buying in stock, it's working out how you're going to manage that risk. And it's different for rams versus replacement ewes, and it's different depending on the diseases you're dealing with. The only thing I would say is that the kind of generic, oh, let's have a quarantine period of 3 weeks is pretty much wrong for every disease. So there aren't really simple rules for these diseases. You have to tackle them one by one in a pretty forensic way. Okay, and once you've done that, it's a case of, again, working with your vet to work out how to manage the risk of these diseases. It takes some time and effort, and the insurance policy approach can be really beneficial to avoid a disaster. I mean, things like boar disease will cause a disaster very, very quickly when they enter a flock. Other diseases can be a slow burning, but very costly disaster if they come in. So just to end on that really positive note, any questions?
Thank you, Piers. Yeah, I've got a couple or two or three here that are stored up, so we'll just run through them. We'll just keep on the Johne's for a minute, if that's okay.
Yeah, sure.
Richard was asking, in Johne's affected flocks, what's the average percentage of animals that you'd expect to be infected? Which obviously varies, but that's the question.
Yeah, it's a really good question. The answer depends on how long it's been there. So I could answer by saying, well, how high can it get? We've got evidence of flocks where 60 to 70% of the animals within the flock are infected with Johne's, but that presumably has been there for a long time. If they've only been infected for 5 or 10 years as opposed to 20 or 30 years, then it may be down in single digits or low double digits of animals infected. So it is that sliding scale, and it'll depend on how the flock has been managed as well. So obviously closed flocks less likely to get it in the first place than open flocks buying replacements, and if it does get in, would take potentially longer for it to work its way through. Obviously, flying flocks as well, if you are unlucky and you buy in replacements every year from a heavily infected flock, you're going to get to that point very quickly. If you're buying from lots of different sources, some of which are not infected, then you'll go up and down in terms of the proportion of flock which is actually infected. But yeah, that's the kind of top end that we've seen it can get to. It may get higher, we just haven't got evidence for that yet.
Sure, thank you. Hope that answers it. And Aaron asked the question a while ago, for the top 3 diseases, Piers, given chronic wasting is a key clinical symptom, what sort of annual percentage of ewes presented this way would you indicate to indicate a potential infection? So in other words, how many ewes wasting away would start to raise your eyebrows to start looking at something?
Yeah, that's an interesting one. I think having a marker of wasting is quite difficult because one person's wasting is not another person's wasting. So I prefer to use targets which are more empirical, so more numbers based. So I would look at replacement rate as the thing that we should be looking at because that's quite a concrete thing. So if you're getting replacement rates in multiple years, which are 24, 25% and higher, I would definitely be worried. And I would definitely be investigating that. Now, individual years can obviously be up and down for lots of different perfectly legitimate business reasons. But chronically at that level, then I would be more concerned. So I would prefer to use that as the kind of metric rather than the more subjective proportion of thin ewes, because people's interpretation of what is thin is quite variable.
Okay, thank you. Hopefully, Aaron, that answers, okay, gives you some answer. And Stephen's asked the question, can OPA spread to cattle at all, Piers?
Oh, good question. I don't think so, no. I'm not aware of any cross-species transmission.
Okay. Hold on. There's a question here from Paul that just popped up. Paul purchased a shearling ram 2 years ago. He's just had him killed and post-mortem done. So 2 years ago, obviously, so young ram. He had gradually lost condition over 6 to 7 months. Not all of the results are back, but it was not Johne's. He was from an MV accredited flock. Which one of the other possible Iceberg diseases could cause us long-term problems if he suffered from them? So
over what kind of period was he losing weight again? Did I say?
I think losing condition over 6 to 7 months, I think.
Over 6 to 7 months.
Yeah. So purchased 2 years ago, so still quite a young sheep, purchased as a yearling 2 years ago. Had him killed and post-mortem done. He's lost condition over the last 6 to 7 months, really, so still only a sort of a 3-year-old sheep. Not all the results are back, but Johne's has been eliminated. It wasn't Johne's. And he was from an MV accredited flock for whatever that's worth.
Yeah, so MV would have been high on the list, so I can understand that. So typically, I think people would regard that as a bit too slow for OPA. So OPA probably would have killed it sooner than 6 to 7 months, I think you said. So probably not that. Boar disease won't give you that presentation. CLA, possible, but it's pretty uncommon. So it would be relatively unlikely to be that. There are, however, a long list of other things which often don't, that they're more syndromes rather than individual pathogens, which would give you that kind of presentation. So we know that chronic pneumonia in sheep is pretty common. Rams more susceptible than ewes. And that can be a whole range of different pathogens, which can give you that chronic what we call respiratory insufficiency, which would give you exactly this chronic weight loss. We also get, cardiac abnormalities, and we had a great one came through the hospital in Liverpool not long ago. So something we see much more commonly in cattle. So issues with the heart valves, and again, this animal showed signs of chronic wasting over many months. So there's a long list of things that can affect them, but a lot of them actually don't have a convenient label on which to hang the diagnosis. So I'm afraid I can't give you a sort of top 2 or 3 likely things to be. I can say something.
Hopefully post-mortems of a histo will actually come back with something tangible, we hope anyway, Paul. So Piers, we've got Clive up in Aberdeen is asking, are the replacement rate percentages referred to excluding standard costs on age, or is it an all-in figure? So I'm not quite sure that I'm getting that.
It's an all-in figure.
Okay.
But mainly because I think what you're getting at is a sort of a standard expectation that you'd expect for each age group. Well, we haven't got that, because I think I don't think we've get a sort of consensus on what that should be. So we took an all-in replacement figure for the flocks involved in the study. Okay.
Hopefully that gives you the answer there, Clive. One from Richard again, with stereomatic production issues, Piers, can any of these diseases be transmitted via vaccination programs?
Potentially. So I wouldn't be as concerned about that as the fact that they're in close contact anyway. If you think about a vaccination run of sheep through yards, the fact they're rubbing up against each other, rubbing snot down the panels as they're going along, that probably represents a bigger opportunity to transmit than actually through the needle, just in terms of volume of infectious material. Because the volume actually in blood and in skin of all of these is very low, but it's the nasal secretions and feces where the highest concentrations are. So for these diseases, I wouldn't be as worried about that. Stereomatic needles and clean administration procedures are very important, in my opinion, but not specifically for these, but for other issues like bacterial hematogenous spread of bacteria causing abscesses in the spinal column. We see that quite commonly where we have dirty vaccine procedures. Not for these diseases per se.
Okay. Thank you, Piers. It's probably worth elaborating. We probably didn't cover it or define it on the Yonis one. When Piers did our initial sort of screening of Yonis within our production, our breeding program, which is 22 odd flocks, we had about just over half, I think, with some level of exposure of Yonis, Piers, which is sort of typical what you'd expect to see in the industry. So I think that's, quite an important factor here, in the fact that what we do, our breeding partners, it's no different to what you see in the industry when you start off, really. And we took the decision quite early on to actually use the Guinea vaccine, and we imported in from Australia, and we vaccinate all the top lambs across all the flocks, be they exposed, showing exposure to Yonis or not, so that all the Nivis rams effectively have been vaccinated with the Guinea vaccine. And we try and do that before they're 4 months old, so that it actually has some sort of effect. We do get the odd case, Piers, where people do post-mortem on rams, for example, and you will get a positive blood test, won't you, if the sheep's vaccinated, possibly?
Yeah, absolutely. So the vast majority of vaccines for diseases in sheep and cattle, you can't distinguish between the vaccine and the wild type infection, so what we call the sort of real infection. The difference you get in the specific case of Yonis actually is that you get a very predictable level of antibodies, which is very, very high. So we can tell pretty confidently when it is a vaccine strain, because the animal's not showing any of the signs in the gut that you'd expect with Johne's, but it has this apparently very high titer, but that's because of the vaccine. So as I said, we're going to talk about the webinar. The blood test for Johne's is really not that useful at all. Even in cattle, it's really not very sensitive. It'll pick up about 30% of actual real cases. So we don't use it that much. What we do use is we use the fecal test, looking directly for the bacteria in the feces. And that is very sensitive and very specific. So it doesn't cross-react or give you other sort of reactions with other potential bacteria. It seems to be very accurate for detecting what we're interested in. So that's the route we normally go down, and that's the route we're encouraging vets to use, rather than the blood test, which doesn't really work for what we need it to do.
Yeah, that's grand. There's a question that's just come in through Messenger for us. Although it's not on the script for this evening, somebody's asking, what could we talk a little bit about CODD and its incidence in the industry and its growing concern? And I think this particular flock's obviously got cod, from what I can understand in the message.
Yeah, well, it is, and there are a list of other really interesting and important diseases that would justify their time. Cod is, well, we know it's been creeping up and jumping up in terms of proportion of flocks infected. The last good study that we had on prevalence is now 5 or 6 years old, and even then it was at 50, 60%. So you can only anticipate it being tracking up probably closer to the 60, 70, 80% of flocks now have it. When it gets into a naive flock, we see what is a pretty typical pattern for a lot of infectious diseases. So you see this big population of susceptible animals. It'll run rife and cause an awful lot of clinical disease until you've got a high level of exposure and immunity to the bacteria. And then it starts to tail off and it drops down, but doesn't disappear, because you're continually getting some animals who are susceptible coming into the flock. Either that's homebred replacements, or that's animals in the mature flock who actually are losing their immunity over time, and then effectively returning to the susceptible pool. So you get this chronic grinding level of lameness. So the numbers can vary a little bit, but I've seen flocks where you've got 20, 25% of the become lame when it first enters, and then it drops down in the second and third year to 3 to 5%, and then to hovering around 1 to 2% thereafter. So that's a fairly typical pattern, and it's something that we'd see actually with quite a lot of diseases, and then it's a grinding problem thereafter. Again, you're going to have different issues if you're a closed flock versus if you're an open flying, in terms of where you're buying stock in from. They could bring it in, and also you could bring in naive susceptible animals. So the two situations will present their own challenges.
Sure, and obviously the basic sort of treatment for normal foot rot doesn't really apply for cod, does it? The same?
Well, yeah, it's a far harder bacteria to treat. So you get limited effectiveness with the antibiotic classes, which are typically used for foot rot. So typically, most people are using oxytetracycline or amoxicillin for treating foot rot. Both of those show some efficacy on some farms against cod, and we do have instances where they are used as first-line treatments. On some farms, they seem to be either completely fail and don't work at all, or don't work well enough given the impact that the disease is having, and the welfare impact is part of that. So use of then the macrolide group of antibiotics is then often the only remaining course of action that people have. And these are decisions with the vet for the farm to decide actually what is the right balance of treatment approaches versus other things you can do to try and slow down the spread. So isolation of clinical cases and breaking up the flock as much as possible to try and stem the spread. That's obviously going to be easier for some flocks compared to others, depending on how you're set up, how many blocks of land you're running over, all of those practical concerns that you have in the way that you manage your flock. So there isn't a simple blueprint, do X, Y, and Z, it'll definitely work, because X, Y, and Z very often can't be done in some flocks. So you have to be able to tailor it to what's possible for an individual flock.
Thank you, Piers. We do have on the cod one, Richard's asking, do we have examples of farms that have successfully eradicated cod? And I suppose I can give you an example of one of our own. We had cod come into our nucleus flock a few years ago. And again, Hamish was very quick off the mark there, pulling sheep out immediately, anything that was suspect. And also, having a specific field that's used for those animals with the treatment, and actually not letting them back into the flock really at any stage. But Piers, you've probably got better examples across the industry.
Well, there are a few examples of that, and there's a couple in the literature as well. So a colleague of mine, Joe Angel, published a paper on cod and on different treatment approaches, and there are a small number of flocks who have managed to eradicate it. It's an awful lot easier if it's a relatively small flock, and you've got the ability to put in a huge amount of time to catch these early cases, because it's all about stopping transmission, and stopping those early cases is absolutely critical. So it is possible. It is relatively rare, and people manage to achieve it, and it's an awful lot easier to achieve if you're small. As Hamish has shown, actually, even in big flocks, it is possible if you really prioritize it, and you can get on top of it that way.
Yeah, and clinically, Piers, it's quite easy to spot, isn't it? The sheep will normally hold 1 foot up.
Well, yeah. So this is a critical issue as to why actually some flocks get it really badly. So it's easy to spot if you've got it in your head that you're looking for it. So we saw an awful lot of flocks. When I was in first opinion practice in the southwest of England, it wasn't really a cod area, and it came into those counties, Cornwall, Devon, Somerset, and farmers there would think that it was just a bad case of foot rot and treat them like foot rot cases until they had gone too far, and then you've got a lot of spread within the flock. But the critical issues you get with it is that you'll get the infection breaking out and giving you this red raw lesion on what we call the coronary band, so where the hairy skin meets the hard horn on the front of the foot. And typically you'll get 1 foot badly affected, swelling, hair loss over that point at the front of the foot. They're the classic signs. It can be confused sometimes by sheep having foot rot simultaneously with cod, but it's looking for this unusual site where you're getting the horn starting to separate, getting this red raw lesion. So it's having that in mind that you're looking for it, not assuming that every lame foot is a foot rot case.
Yeah, thank you, Piers. While we're on examples of flocks, we have an anonymous attendee who's would like to know, do we have any advice on managing flocks with OPA, and is there a current research being carried out on developing better screening for the disease? I can give you a bit of a heads up before Piers, I suppose. We have identified very low levels of OPA in 3 of our ram breeding flocks. And again, we've identified them through post-mortem, as Piers said. So being very vigilant on our cull ewes, trying to understand what's going on with them. One of the key things, because we have all the pedigree information, is if we find one on post-mortem, we track down the progeny straight away and pull them out of the system. And that's proving to be quite useful. And then it's very much monitoring condition scores, although again, we don't really see sometimes animals that go down that quickly in condition score. They tend to die, but we've got very low levels under 1%. We have done some strategic screening, thoracic screening, as Piers identified before. So we use all the tools available to us, really. But we put the sheep under quite a lot of pressure all the time, and anything that isn't coping with that pressure, gets pulled out into a management group, it's given a slightly nicer environment to live in. And if it doesn't recover from that very quickly, we actually put it into an at-risk group and identify that. But Piers, again, I'm sure you've got much better examples in industry than that one. But
yeah, so a true screening test needs to have very, very high sensitivity, so that you're finding all of the, or as many as possible of the animals who are actively infected. And to do that, really, you have to have a lab-based test, because all of the tests that you could imagine with imaging, so whether that's ultrasound or any other imaging technique, you're only visualizing things which are big enough to see, by which time virus has obviously been there for some time and potentially spread. And with ultrasound scanning, you can't actually scan the whole of the lung field. So you're not even scanning the lungs. So it's got obvious limitations of screen test that way. What we really need is a lab-based test which looks directly for either the virus or an antibody response. Antibody response is really, really difficult with YARC-CT, with OPA. There is a test available on tissues, which is offered by APHA, which is used as can be used as a confirmatory test for post-mortems. That would be a route, better optimization of that kind of test for something which should then be applicable as a screening test, where you could then retest the animal if it was inconclusive and take that approach. But it isn't particularly sensitive either at the moment. But that is where we would normally look for appropriate screening methodologies, screening type tests. It hasn't been an area that's been well funded, typically because it's actually a disease which is relatively uncommon. So it hasn't been something which has attracted a lot of funding compared to the other diseases we've talked about. And there's that cost benefit impact that you've got to think about as well. So compared to some of the things that we've talked about, it probably doesn't cause as much production limiting effect in most cases. It may well be an instance where actually you do see that this pattern that we're talking about with CODD, where you get very naive flocks where there's a high proportion of susceptible individuals, then it runs rife until those susceptible genotypes, those susceptible individuals have been removed, and then it drops down to more chronic levels. We certainly do have a lot of farms that we know of which have been managing it for a long time, and just doing it by promptly pulling out animals which aren't coping. So there's lots of examples of that. But yeah, it's not something which I think has attracted a lot of funding in the past for those reasons. So yeah, hopefully that answers the question.
Yeah, and there are commercial examples, Piers, isn't there, where the local vets have done some thoracic scanning and have instigated a cull on those sheep that have got some masses there. And in a commercial scenario, doing that en masse obviously remove sheep. There's probably, as Piers said initially, a risk that you're moving sheep, getting rid of sheep that are actually going to be fine because you're misdiagnosing what's there. But as a tool, it's still getting rid of, sheep that have got some noise going on in their lungs at that particular moment in time.
Yeah, and it comes down to a value judgment as to how many incorrect diagnoses do you think is good value. So you could theoretically have a test which killed 50% of a group of sheep in the effort of finding 1 or 2% who are actively infected. And you could argue that would be cost effective if the disease is massively, massively damaging. That degree of collateral damage of misdiagnosing false positives, you could argue would be effective. I think what I would urge any vet or any farmer to do is, when you're thinking about potentially using something subjective, like ultrasound screening, use it for screening, and have that conversation with the person doing it to understand actually what are the likely number of false positives, because a lot of people would assume tests are better than they actually are. And going into it with unrealistically high expectations can be quite damaging, especially when we've got instances where we find that actually 50% of the apparent positives are actually not positives at all when we post-mortem. Some instances even worse than that. So more false positives than real positives. So there's that element to it as well, and as well as the element I said before that it's pretty unrealistic to think that you're going to be able to eradicate it just because you're not able to detect it earlier before it's almost certainly already spread. So as long as people have realistic expectations and they think it's appropriate for their particular situation, I think that that's absolutely fine. That people have to go into these things with their eyes open and be realistic, because it is a very different proposition to any of the other validated, repeatable tests that we can do with lab assays, which are objective, not subjective, and which can be repeated or confirmed with other complementary tests. None of that's possible for OPA at the moment. Yeah, I think that leaves us in a situation where we've got to be really quite cautious.
Yeah. Thanks, Piers. Aaron, a very good question here. Strategically, does setting up a CUL-U Iceberg monitoring testing program with your own vet represent a good return on investment, even if your replacement rate is within the normal parameters and your fertility, etc., is within normal parameters? So in other words, Piers, even if your performance is up there in terms of what you'd expect, is actually doing this something sensible or not?
I suppose that's a question about how useful would it be to identify a problem at a very early stage if it arises. So a problem at a stage before it reaches a noticeable level in those metrics. So that comes down to a value decision by the farm, really. I don't think there is legitimately a simple answer to that, because whether it's good value for money would depend on how much it's actually going to cost you. So how much it's going to cost you for the vet or post-mortem service to do that, versus the value of the CUL-Us at the time for your generating flock, and the potential diseases which you don't currently have that you might want to be able to screen for through that approach. So it's a big maybe answer to that, I'm afraid.
Sure. I think obviously it makes absolute sense, doesn't it, to understand what the age profile of a flock is and whether you're actually getting, high cull levels and sheep not lasting. And, on that graph that Pierre showed was very graphic in showing your replacement rates, the implications of that. And of course, we're also busy farming most of the time that quite a lot of those things pass us by. And we're getting into that scanning period now where, we hope for a good scan, but there's also losses at different stages after that, Piers, isn't there? But you use quite a lot of that production data to actually hone in on disease, though, don't you?
Yeah, absolutely. I mean, there's a vast amount of data that sheep systems have that they don't realize is potentially valuable for analysis. And it's a massively wasted opportunity for most people. I mean, if you compare the level of analysis of metrics that we would typically do in a pig system, poultry system, or even in a dairy system, there's actually analogous things that we can look at in the sheep system, which we don't routinely do, even though the data is there. And some of that is around how available that data is for processing. And some of it's around people just not prioritizing it in busy lives that we all have. But even simple things, like we mentioned, the creep consumption there. I mean, that's been a really nice example. A couple of very nice clinical case workups done by colleagues have shown that to be a really interesting metric. Again, the distribution of sale weights and timings to sale for the lamb crop is again a very useful metric to understand how that distribution looks, because you want it obviously to be as tight as possible with short a tail as possible. All of these things we can look at, but typically we don't, because we don't either make time for it or we don't make the data suitably available to be able to do it easily.
Yeah. Thank you, Piers. Hopefully that answers questions for people. The other key message I think you've made this evening is that, whilst the recommended 3-week quarantine may help towards, certainly quarantine dosing resistant worms, possibly looking for things like scab, looking for lameness, when it comes to iceberg diseases, we have to go to the source flocks to understand what's happening at that level, don't we? Be it a rab breeder or a ewe breeder. And I think that's a very important factor for you all to take on board, that you need to have that discussion with the people you're sourcing your sheep from in a very open and constructive way to understand, you know, what's going on at their flock level, because you are by default buying their health status. And that's really quite important for people to understand that, I think. So Piers, I think that's us for questions. I think people probably, hopefully you've all had something out of this evening. Laurie will send the recording out to everybody that have been on and those of you that haven't. We have been asked by one of the Aberystwyth ag students who's doing a dissertation on iceberg diseases. Would we circulate the questionnaire? And I think Laurie will send that out to you when she's sending the recording. So you'll see a link to a questionnaire from one of the ag students in Aberystwyth. So obviously it's entirely up to you if you want to fill it in, but she's gathering quite important data, I think, to understand attitudes and behavior towards these sorts of things. Piers, thank you very much. For answering all our questions as usual in comprehensive, measured and informed way. And we're very grateful for all your input in what you do with us. Hope you've enjoyed it all, and the next webinar is in 2 weeks' time, where we're talking about actually reducing the carbon footprint of your sheep system. Ours, yours, collectively, how we go about doing that. And we've got Suzanne Rowe, who's one of the initial scientists in New Zealand that started the whole reduction in methane and selection speaking, and she's very, very good. So yeah, those of you who can make it in 2 weeks' time, please join us and we'll be talking carbon. But for now, thank you, Piers. Thank you all. And good evening. We'll start.